Now, let's begin our lesson. Practice Chapter 3, Hemodynamic Disorder. Today, we're going to observe the two slices. Also, we will observe these gross specimens. Slide 1. Chronic pulmonary congestion. First, let's review the histological structure of the lung. This is a small bronchus with gland and cartilage. This is a branch of the pulmonary artery that goes with the bronchus. This is a bronchiole with a smaller diameter. Here and here are the terminal bronchioles. Here is the respiratory bronchiole. This is alveolar duct. The rest are alveoli. This is the alveolus. The wall of the alveolus is very thin, covered with a single layer of alveolar epithelium. The alveolar epithelium is composed of type 1 and type 2 cells. The adjacent alveolus are close to each other, only separated with a thin layer of connective tissue. The septum contains capillary, macrophages, and other components of connective tissue. In alveoli, macrophages also can be observed. Now, let's see the slides. Chronic pulmonary congestion is caused by left heart failure. Small vessels, most of them are capillaries, also include some small veins in the septum, are dilated and engorged with red blood cells. The septum becomes thickened. The pink edema fluid, also called blood plasma, can be seen in the alveoli. Intra-alveolar hemorrhage also can be observed. That is, red blood cells may appear in the alveoli because of the rupture of small blood vessels. The most important feature for this slide is the presence of heart failure cells in the alveoli. The heart failure cells are also called hemosiderin-laden cells. There are many coarse brown granules located in the cytoplasm. Here shows aggregation of heart failure cells in alveoli. Brown granules in cytoplasm can be dyed blue by Prussian blue reaction. Here, the alveolar septum becomes thickened and fibrotic. Now, do a summary. As for the chronic pulmonary congestion, in the alveoli, edema fluid, red blood cells, and heart failure cells can be observed. In the alveolar septum, capillaries and small veins are dilated and engorged with red blood cells. Septum become thickened and fibrotic. Now, next slide. Hemorrhagic pulmonary infarction. Here, this wedge-shaped part is infarct area. This part is viable tissue, that is lung-infected tissue. Between them is an inflammatory response zone. The infarct area, it contains coagulative necrosis and hemorrhage. All nuclei disappeared within the infarction, but an outline of the tissue architecture is still recognizable. That is, we can still recognize where are the alveoli, where are the septum. We call this as coagulative necrosis. Alveoli are filled with red blood cells. We call this as hemorrhage. The wedge-shaped infarction has the features mixed with coagulative necrosis and hemorrhage. Here is non-infected or viable tissue, which displays the feature of chronic pulmonary congestion, a narrow rim of the inflammation around the infarction, which separated the two parts. We call this as inflammatory response zone. Here is infarction of lung tissue. Here is non-infarction. This is inflammatory reaction zone. Here it is the infarct tissue. At high magnification, all nuclei disappeared, but outline of areas and septum still remain. We call it as coagulative necrosis. The alveoli are filled with red blood cells because of hemorrhage. This shows inflammatory response zone, a narrow rim of the inflammation around the infarction. It contains newly formed capillaries, inflammatory cells, and fibroblasts. Indeed, it is granulation tissue. Here is non-infarction. It is zone of chronic pulmonary congestion. Heart failure cells can be observed. Now, do a summary. As for hemorrhagic pulmonary infarction, we can observe infarcted area mixed with coagulative necrosis and hemorrhage. Lung-infarcted area, the main feature is chronic congestion, and inflammatory response zone separates the two areas. Now, let's observe gross specimen. This is chronic pulmonary congestion. We call it as bronch induration of lung. See next picture. Volume of lung reduced, and the capsule shrank. Sponge-like appearance disappeared and became solid and firm. Lungs are rusty brown. There are many heart failure cells aggregate to form visible brown spots. This is chronic liver congestion. Because of chronic right heart failure, hepatic vein outflow was sluggish or obstructed. Blood accumulates in hepatic sinusoid, showing the liver overweighted, enlarged, with tensed capsule and blunt edge. Because the centrilobular congestion with hepatocyte necrosis, central veins dilated, sinusoid is full of blood, shown in cut surface in central lobule. It shows zones of red. However, in peripheral lobule, hepatocytes undergo fatty degeneration, so in cut surface, zones of yellow can be observed. Here is the representative area in one liver lobule, zones of yellow alternating with zones of red. Many hepatic lobules here had the same shape, so nutmeg liver formed. This is a long thrombus in the vein lumen. Here is the head of thrombus. This is body. This is tail. The head is mainly composed of platelets and fibrin. It is gray-white in gross observation, and the tail is mainly composed of red blood cells, so it is dark red. The body contains platelet, fibrin, and erythrocyte rich, so it is white and dark red mixed. Because fixed in formalin, it is white and black mixed. The thrombus is more closely attached to the wall of blood vessel and is not easy to fall off. This is another thrombus in the vein. Question: what clinical consequences that thrombus will bring about? This is mitral valve. The valve is damaged. There are several grayish yellow vegetations on it. The vegetations included platelet, fibrin, leukocytes, and bacterial, mass, and necrotic tissues. These vegetations are fragile and easy to fall off and reach other places, leading to embolism, infarction, and other consequences will be caused. As time goes by, at the bottom and edge of vegetations, granulation tissue grew up and led to organization, fibrous connective tissue formed. finally led to deformation of valve. This is spleen. Here is a wedge-shaped area. It is coagulative necrosis. It was usually caused by the loss of blood supply, so the necrosis is also called anemic infarction. On the cut surface, the infarction is firm and pale. The apex toward the hilum. The base is located in the capsule. The infarction is surrounded by a rim of inflammatory hyperemia zone. Here is lung infarct area. The inflammatory hyperemia zone forms the clear boundary separating the two parts, infarction and lung infarction. This is a hemorrhagic pulmonary infarction. Here is a dark red, wedge-shaped infarct area with the apex towards the hilum, and the base is near the pleura of the lung. Question: what are the changes in other lung-infected areas? Most infarcts are ultimately replaced by granulation tissue and scar formed. Today we have learned some slices and gross specimen. Here is a case. Please answer the question. This is the answer. Okay, that's all. Bye-bye.